Good afternoon, ladies and gentlemen. Welcome to the WALS lecture. Those of you who braved the conditions to be in the auditorium today, and those of you who thought your desks would be a cozier spot. It's my pleasure this afternoon to introduce our speaker, Stuart Orkin, who is the David G. Nathan Professor of Pediatrics at Harvard Medical School and Chair of the Department of Pediatric Oncology at the Dana-Farber Cancer Institute, as well as a Howard Hughes investigator. He has for many years been one of the preeminent figures in the study of hematopoiesis and hemoglobin disorders. After graduating from Harvard Medical School, Dr. Orkin began his research career here at the NIH in the laboratory of Dr. Philip Leader, where he studied properties of erythroleukemia cell lines. That interest expanded upon his return to Harvard, where he began to examine globin gene organization in the alpha and beta-thalassemias and devised improved assays for prenatal diagnosis of the sickle-cell mutation. In the following years, his group published many papers dealing with the thalassemias and sickle-cell anemia, and increasingly with ways to treat these disorders. Chemical methods were unsatisfactory because they have undesirable side effects. Approaches that were used in the field increasingly centered on reactivation of the fetal globin genes, which would replace or complement the mutated adult globin gene. As Professor Orkin understood quite early on, the best solutions were likely to arise from an understanding of globin gene regulation. The cloning by his laboratory of GATA1 was an important step in this direction. GATA1 is a master gene that regulates most of the events that produce red cells from stem cells in bone marrow. Since then, Dr. Orkin and his collaborators have cloned other genes involved in red and white cell development and studied their function in recent years, increasingly in hematopoietic stem cells. His constant pursuit of the goal of reactivating fetal hemoglobin was rewarded in 2008 with his discovery that BCL11A inhibits fetal globin production, beginning a widespread effort in the field to target BCL11A. This and related approaches may soon provide effective methods for treatment of sickle cell disease and beta-thalassemia. Although a large part of Dr. Orkin's work has been devoted to these problems, he has also made important contributions to understanding the mechanisms that maintain pluripotency in embryonic stem cells and an analysis of various cancers, such as bone and breast cancer and cancers of the hematopoietic system. Importantly, Dr. Orkin's influence and impact derive not only from these major accomplishments, but from the large number of his students that have themselves become distinguished investigators and contributors in the fields of hematopoiesis and stem cell biology. Our understanding of hematopoiesis would surely be much less complete without Dr. Orkin's work, and we welcome him back to the NIH where he started.
Thanks, Anne, for the kind introduction. I have a post-viral cough, so if I cough in the middle, just ignore me. I'll get through this. So what I'd like to try to do is tell you what we've been doing recently in trying to understand fetal globin switch and what we might be able to do about it. And this really traces the last probably 6 years' worth of work in one aspect in the lab. And it really does begin here while I was at the NIH as a research associate with Phil Leader in Building 6. I began studying red blood cells and erythroleukemic cells and differentiation, not knowing that this would actually be a 3-decade-long process. Actually, I thought it would only be for 2 years at the NIH, but I've continued. So this is standard view of hematopoiesis in the mouse, in which there's transition from mouse yolk sac to fetal liver, ultimately to the bone marrow during development. And obviously it's a very dynamic system, and there's tremendous interest in trying to understand what it is that allows the stem cells to renew and progenitors to develop at each stage. But for the purposes of this talk, I'll be focusing really only on this portion of the problem, and that is how it is during red cell development or development in either mouse or human. We actually switch in the human from embryonic to fetal to adult beta-globin. And we've known the structure of the locus, the beta-globin locus on chromosome 11, for more than 30 years. And in fact, we've had the DNA sequence of the entire locus for more than 30 years, none of which really gave tremendous insight into this switch. And this switch is important because the disorders of the beta chain, beta-thalassemia and sickle-cell anemia, make one dependent really on the beta chain in adult life. And the gamma chain, which is part of fetal hemoglobin, is silenced, but not to zero. It's silenced to a couple % in the normal adult individual. So we've been interested in how red cells, as well as other hematopoietic cells, are made, how these cells are programmed from hematopoietic stem cells, how do they go awry in disease, either non-malignant or malignant disease, and what can we do about these disorders once we learn something at a basic level. Now, in terms of thinking about fetal versus adult globin gene expression, one can ask, well, why study it? Well, first, it's really a paradigm for developmental stage and tissue-specific gene expression. The genes are expressed in a tissue-specific manner, but also in a developmental-specific manner. But why should we really care? And there are two obvious reasons from a disease standpoint. One is sickle cell disease, which was first described more than 100 years ago by this cardiologist, James Herrick, and where he first noticed the sickle cells. Linus Pauling called it the first molecular disease, and then Vernon Ingram identified the single amino acid change that defines sickle hemoglobin. And then there's beta-thalassemia, which is a disorder of production of beta-globin, not the structure of the beta-globin itself. Now, in terms of sickle cell disease, we've known the mutation for more than 50 years, yet I think it's sobering that none of this information really has any bearing on current therapy. I think that's a statement of either the challenges involved or perhaps where we need to go. In terms of thinking about the hemoglobin disorders, particularly sickle cell disease, in this country there are about 100,000 individuals, but worldwide there are many more. On the African continent, there are about 300,000 new births per year. And it's predicted over the next several years that there'll be upwards of 5 to 6 million children with sickle cell disease on the African continent. And if one assumes that there'll be better control of malaria, HIV, TB, children who die young with sickle disease are going to live longer and present even a greater challenge to the health system. So this is a global problem which is not going away at all. We've known for many years that if one's fortunate enough to have higher level of fetal hemoglobin, if one has sickle cell disease or thalassemia, one has less severe disease. This is from the natural history study of the NIH by Ora Platt, showing that higher fetal hemoglobin predicts a greater survival in sickle, and this is true in thalassemia as well. What level would be curative? What level of fetal hemoglobin? Here the cutoff is 8.6%. Think somewhere on the range of about 15 or 20% fetal hemoglobin, if distributed in all cells, that is pancellular, would have a tremendous beneficial effect. The only approved agent for sickle cell disease is hydroxyurea, which is effective at reducing pain. It has issues that make it impractical for third world usage at high frequency. It's not as effective as transfusions, though, at preventing recurrent strokes in children. It's not a perfect drug, and it's sort of a halfway measure at this point. It is changing the natural history and really getting children through through childhood years and really presenting adult physicians with patients with sickle cell disease with intended consequences later on. So what do we do now? Basically, we focus on the consequences of the sickle mutation: anemia, pain, stroke, pulmonary disease. Where we'd really like to be is up here, to focus our therapy at that point. And there are really only two ways we can think of doing that. One is correcting the sickle mutation, perhaps with gene editing techniques, which at this point, and I think for the foreseeable future at least, is not practical at the efficiency that's needed. Well, the alternative is to somehow prevent sickle hemoglobin from polymerizing and sickling. That may be feasible with small molecule design, or alternatively increasing the level of fetal hemoglobin, essentially to replace sickle hemoglobin, or in the case of beta-thalassemia, replace the deficient hemoglobin and allow for better hemoglobin formation in the cell. So this is again the problem, the switch from gamma to beta. How do we understand it? What do we know about it? There are really sort of two issues that I think one has to distinguish really at the outset, which sometimes get confused, and that is distinguishing fetal versus adult gene expression in erythroid cells. There are really two aspects. One is fetal cells are different than adult cells. They express about 1,000 different transcripts. So there's something different, there's a fetalness to the cell. And the other is what kind of globins produce, whether it's fetal globin or adult globin. You might think it's the same problem, but we actually think at a transcriptional level, at least as far as we understand it, they're really, for the most part, distinguishable. The sort of fetal program, the whole developmental program, is really regulated through a set of stage-specific enhancers in genes that are expressed in a erythroid-specific manner. Whereas our data to date suggests that the control of the globin genes is largely more locus-specific and really independent of this fetal stage. So in other words, when one talks about reactivating fetal hemoglobin, we're really talking about reactivating the fetal genes, the gamma genes, and we're not talking about driving the cells back to a more fetal state. And sometimes that gets confused. One of the systems that's very convenient, it has its flaws, but it's very convenient for the studies I'll describe, is the use of primary CD34 cells, which can be easily mobilized in adults, can be expanded, and then differentiated to form erythroid cells. These are the cells used in bone marrow transplantation. One can obtain these either from adults or from fetal liver, where one can have fetal cells or adult cells, and the fetal cells retain their fetal globin gene expression and fetal program, shown here with expression of gamma, whereas the adult cells will express the adult. And we can use this system for biochemistry, proteomics, gene expression, ChIP-seq, any sort of technology that will allow us to distinguish what's different about gene expression and the control in these states. I'll just summarize what we've learned about the fetalness aspects, because that's really a separate topic. This is one way of looking at sort of the way cells establish their fate. We have epigenetic regulators as well as intrinsic drivers of differentiation, which define the cell identity. And what we found for the most part is that the fetalness, the stage specificity, is programmed by stage selective cofactors. It's not programmed by the master transcription factors for the lineage. It's programmed by accessory cofactors that are developmentally regulated themselves, but may be used widely in other settings. Examples of that would be MIB, which is essentially an adult-specific hematopoietic factor, or the interferon regulatory factors, which surprisingly contribute to the adult phenotype as well. And what we've learned is that the stage specificity is largely attributable to these enhancers and the factors that interact with them. And for the most part, these are unrecognized cofactors, or previously were unrecognized. And I think there's still more to be discovered. The mechanism accounts for the overall stage specificity, but really doesn't provide tremendous insight into the globin locus per se. And the expression of the fetal or adult globin is not coupled to this stage specificity in a strict manner. So what do we know about the control of fetal versus adult hemoglobin? This is one of the first rational descriptions in the literature, again from Vernon Ingram in 1961, in which he imagined that one switched down the expression of the gamma chain and switched on the beta chain, while the alpha chain is constant. This sounds simple today, but prior to this in the literature, there were rather confusing models because at the time it was unclear how many genes there were that encoded these proteins, even what the organization of the genes was. There's a long timeline to understanding this switch. There were genetic studies of families largely in this period between the 50s and the 70s, where one really defined that hemoglobin F was a inherited trait, largely controlled at the genetic level. There were families that had increased fetal hemoglobin. Some of the mutations became identified thereafter in the ensuing years. The globin genes were first cloned around 1980 with DNA sequences obtained soon thereafter. That information was not particularly helpful in terms of understanding the switch per se. We identified in Gary Felsenfeld as well GATA1 just before 1990, and there have been lots of other studies in intervening years, but for a while we were kind of stuck on the problem. We were kind of wandering in the desert and didn't know where to go. And I think the field sort of came to a halt until about 6 years ago with the introduction of genome-wide association studies, which actually did provide the first genes that regulate fetal hemoglobin in adults. Probably most of you are familiar with the way these studies are done. One assembles groups of individuals, populations, either patients or non-patients or different traits. One types their DNA with single nucleotide polymorphisms across the genome and attempts to correlate it with particular genomic position. This has been done many, many times over with diseases or traits. This is one of the NIH websites you can go to. For many of these, one identifies a locus, but the locus is still obscure as to how it impacts the particular entity. The situation here is a lot simpler, we believe, and we can actually trace things down, as you'll see, essentially to the nucleotide level. So genome-wide association was performed, measuring either hemoglobin F itself or F cells, which is a measure of cells that contain increased amounts of F. And these were done both in the UK by Suile Thien and in Sardinia by Antonio Cao and their colleagues. We collaborated with Antonio Cao's group. And there were 3 hits in the so-called Manhattan plot. And this is Manhattan with the Empire State Building focused on B11A, which is the only thing I'll concentrate on for this talk. There were 3 hits, one at B11A on chromosome 2, the MIB locus on chromosome 6, and the beta-globin locus. It was already known that genetic variation in the beta-globin locus leads to different hemoglobin F levels. Many cases not understood why exactly. MIB had been identified actually through linkage studies prior to this, or at least that region of the genome. And B11A was sort of the new kid on the block. And if one sums the contribution of these, at least 50% of the genetic variation hemoglobin F roughly is accounted for. So these contribute to a large percent of the genetic variation, which is atypical for genome-wide association studies. And also the other sort of curious observation is that this variation occurs across all populations. It's not limited to particular ethnic or geographic groups. It's worldwide distributed. It's common variation. So we've been studying B11A in the intervening time. This is what the original data looks like on the NIH sickle cell cohort at B7A at one SNP. Here are the low hemoglobin F individuals with a mean of about 4%. The highs have about 11%. There's a lot of scatter in the data, so it's not predictive in any way at a single individual. But you can see that there's a difference of about 4 to 11%, almost threefold difference in the basal hemoglobin F level, dependent on the genotype at the B7A gene. And we'll come back to this later as to what this means in terms of B7A per se, because I think it's important thinking therapeutically. Everything we've learned in the ensuing 5 years or so, says that B7A is a quantitative regulator of hemoglobin F. If you increase the amount of B7A, you decrease the amount of F, and conversely, if you decrease the amount of B7A, you increase the amount of hemoglobin F. I'll just summarize some data that's already published that supports this, and the general notion that B7A acts directly within the locus. By chromin immunoprecipitation in adult erythroid cells, it interacts with the LCR, the major control element. It's bound over the silent embryonic genes, and between gamma and delta in this region, which is associated with deletions that are known to yield higher F in individuals that have been known for many years, these deletions. Curiously, it does not bind over the fetal genes, so it doesn't repress the promoters directly as best we can tell. We think it acts in some long-distance manner. If you take those CD34 cells I described, inhibit expression of the protein with shRNA, you induce gamma globin RNA and fetal hemoglobin. This is not enormously specific. Many agents will do this for a variety of reasons in these cells, but it's at least reassuring. If one uses a mouse that has B7A gene knocked out and incorporates into that mouse a human transgene that expresses the human cluster in a developmentally appropriate manner, at least in the mouse, what happens is the gamma gene, which is normally expressed, is shut off around birth in the human, but is shut off in the fetal liver in the mouse, is no longer silenced appropriately, and the heterozygote is in between, so it's dosage sensitive. And I should just mention, for those who aren't aware, the mouse has no fetal equivalent. The mouse goes from embryonic to adult. So the human fetal gene is sort of is an unknown commodity to the mouse. It has to be handled either as an adult or an embryonic gene. It's handled as an embryonic gene. And finally, if one uses a conditional mouse, allows the human transgene to be repressed, and it's repressed about 2 orders of magnitude greater than in us, so it's about 0.1% or less of the total beta-like RNA. If you then remove B7A conditionally, you get about 500 to 1,000-fold induction of gamma RNA to a fairly substantial level. And all these data suggest that the switch is reversible and is largely mediated through B7A in this context. There are many other proteins that have been described in the literature as affecting hemoglobin F or globin gene expression. So one can ask, well, how important is any single protein? And probably the closest we can come to this as an experiment is to use the existing mouse sickle cell models in which the mouse genes, the globin genes, are ablated by homologous recombination, and human genes have been introduced that are developmentally expressed. And one can generate then a sickle mouse, and the mouse is anemic and has all the sort of consequences and challenges of a sickle cell individual, renal damage, anemia, etc. And one can then ask, if one removes B7A, in this case in an erythroid-specific manner, to avoid any other consequences, just takes down erythroid cells, what's the phenotype of this mouse? And the result was actually quite striking to us, much cleaner than we might have anticipated. This is the control mouse. Here's the sickle mouse with the sickle forms. And here's the cross in which we remove B7A in the erythroid cells. The standard hematology is entirely normal. The retic count, a measure of young cells, is brought back almost to normal. And the urine concentrating ability, a measure of renal damage, is essentially normal. indicating that these mice are phenotypically normal, and we've had them for 2 years after, and they're perfectly fine. And why are they well? Well, they express a lot of gamma RNA, not surprisingly, and the fetal hemoglobin is distributed in most of the cells, so-called pancellular expression. About 85% plus of the cells contain lots of fetal hemoglobin. One curious aspect of B7A is that if one removes it in the red cell formation and production is entirely normal. So this is a transcription factor, as best we can tell, controls very little else that's critical to a red cell other than the globins. Virtually unheard of in our experience. So this is really what we've come to then. We've come to a situation in which we can imagine a sickle red cell containing mostly sickle hemoglobin, a small amount of hemoglobin F. We have a target B7A, and we'd like to think about somehow inhibiting that target and generating a cell that has more hemoglobin F, not necessarily 100%, but enough hemoglobin F to prevent sickling, and hemoglobin F is a great inhibitor of the sickling process. And one nice feature is there's no resistance. It's unlike a cancer target where there'll be some resistance mechanism that will circumvent your inhibition. The cell really is sort of agnostic as to what kind of hemoglobin it actually expresses. And so we imagine there's some therapeutic level as we decrease B7A concentration, generating more hemoglobin F, and obviously at some point we'd like to be in this range. And we'll come back to that, where it may be. Now I've skirted over one major issue, and that is the GWAS actually identify the gene. So this genetic variation in the B7A locus somehow ultimately affects the expression of the hemoglobins. And so how could this be? Well, one easy solution is there could have been a change in the structure of the protein, such as B7A protein has different function. Just suffice it to say the protein is normal, and all the genetic variation occurs within a large intervening sequence within the gene. And this is a depiction of the B7A gene. Here's a large intron, and the chromatin profile in erythroid cells within that intron has the hallmarks of a regulatory element, marked with H3K4 monomethylation, H3K27 acetylation, binding of GATA1 and TAL1, two master transcription factors in 3 regions. This is in human cells. This region is not marked in the brain or B lymphocytes, which also express B7A, and which B7A is important for, or in T cells. And all the sentinel SNPs, the highly correlated GWAS SNPs, lie within this region. In addition, as a regulatory element, this region is marked by DNase hypersensitivity. That's erythroid specific. It's not found in other lineages. And basically, if one blows this up then, we have 3 DNase hypersensitive sites in the human, with the promoter to the right at plus 55, plus 58, and plus 62 in the gene. And this is a putative regulatory element. We knew before this that B7A is exquisitely expressed in a developmental specific manner itself. If one looks at mouse cells, primary primitive cells, that is from the yolk sac, have no B7A. They're expressing embryonic globin. As soon as the cells develop in the fetal liver that express, in the case of the mouse, adult globin, you see high-level B7A expression. This is a Western blot, and you see it in the bone marrow. The human is different in that in the primitive cells, the human yolk sac cells, the embryonic cells, there's no B7A. But in the fetal liver, where the switch is just beginning, one sees a small amount of B7A and much greater amounts in the adult bone marrow, showing that there's a developmental pattern of expression, but it differs between human and mouse. In mouse, it's really kind of on-off, and in the human, it's off and then gradually comes on. And one of our predictions, which we haven't yet proved, is that this developmental difference may be responsible in part for the difference in switching between mouse and human. and we're trying to test that. But the gene, the BCL11A gene, is developmentally regulated in a erythroid manner. And in fact, this regulatory region between plus 55 and 62, if one takes this region out and links it to beta-galactosidase as a reporter with a minimal promoter and puts it into a transgenic mouse, the transgene is expressed in the fetal liver, where BCL11A is first expressed, but it's not expressed in B cells, which express normally high levels of BCL11A, so this regulatory element is not one for B cells. And it's expressed in the fetal liver, but not in the peripheral blood at that time, which have circulating primitive cells. So in fact, this is again an enhancer then that's erythroid and adult stage specific. We went back with our genetic colleagues to look at the SNPs from GWAS, and there's one SNP, this one, which is actually located in the plus 62 element, which is most hardly correlated with hemoglobin F if one does the appropriate conditioning of the data. And in fact, that we believe is a causal SNP. That particular SNP changes sequence within this composite site, which in the field is known as a composite tau GATA1 site, which is a known regulatory element in hematopoietic cells and in erythroid cells. And in fact, this SNP is associated with a change in allele-specific chromatin binding by GATA1 and tau1, such as less binding on the variant allele, and there's actually less RNA expressed from the variant allele, indicating that this is really a causally associated SNP. But the important finding is the difference of expression between the normal allele and this variant allele is about 40%. So that tells us that the high F allele puts out about 40% less BCL11A than the normal allele. And just remember back to the original GWAS of the sickle going from 4% to 11%. We think that this sort of accounts for it, roughly that order of magnitude difference. So in fact, the genetic variation occurs within an enhancer that specifically affects expression in erythroid cells. And this really fits the emerging theme, a theme that was emerging at the same time, that most of the GWAS hits in disease or traits occurs in non-coding DNA, and most of this is actually associated with putative enhancers or regulatory elements. So this is a specific example of that. But how important is the enhancer? Natural genetic variation has a modest effect on expression, maybe 40%. Is this akin to the small effect size of many GWAS? One of the sort of criticisms of GWAS studies is the effect size is rather small. So how could the gene be important? Well, it's what natural variation gives you, and the modest effect may be restricted in a sense by natural variation. So what's the true role of the enhancer? Could it be larger? And in order to get at that, we actually turned initially to the mouse. We've turned to human cells as well. But we looked at the homologous region in the mouse, which is depicted here in erythroid cells. It's, in architecture, it's overall very similar, but in detail, actually, it is different. The central hypersensitive site is less apparent in the mouse than it is in the human. And we used genome engineering to remove the enhancer. Now, I'm sure most of you are familiar, this is a rapidly moving field. The field started with conventional gene targeting, which is of low efficiency and not really applicable for clinical use. It progressed to the use of modified zinc fingers and talons, which have high efficiency, which actually, in case of zinc fingers, are actually in clinical trials in HIV. And more recently, the explosion on CRISPR-Cas9 as a very high efficiency, easily used system for gene editing. At the time, which was actually a few years ago now, we first used a pair of talons to remove the regulatory element from mouse erythroid cells, and we did that in MEL cells. And if you do that, the expression of BCL11A goes essentially to zero. It's less than 1%. The protein is absent, And concomitant with this, one sees about 100-fold induction of the embryonic globins in these cells, which is exactly what we had seen from other kinds of studies. So in fact, this enhancer is really obligatory for erythroid gene expression. Natural variation modifies expression a little. Taking the enhancer out, one essentially has on-off. If one does the same experiment in B cells, which express about 10 times more B7A normally, and B7A is essential for B cell development, one sees no change in B7A expression or protein. Again, saying this is a tissue-specific element, and the B cell elements reside elsewhere. So essentially, in the low hemoglobin F situation, the normal situation, we produce B7A protein, represses the fetal genes, and in the case of sickle disease, one has standard disease. In the case of natural variation, one could reduce the amount of fetal hemoglobin, the B7A expression, modestly, maybe 40% roughly, and this will alleviate hemoglobin F repression a little, but really doesn't have a major effect on sickle disease, at least in the individual. But what we're speculating and really proposing is that if one removes the enhancer, one would have no B7A, maximal fetal hemoglobin production to a level that we still have not yet determined, and one would relieve the sickle phenotype. So this is the clinical experiment that really is derived from these data that we hope will be performed in the not too distant future, and that is to collect hematopoietic stem cells, use sequence-specific nucleases to disrupt the enhancer. One could also talk about disrupting the B7A gene, but we can discuss that later as well, and reinfuse these to basically create a genetic cure of the disease. One reason we may not want to remove the B7A gene itself is it has other roles in hematopoiesis. It's important in B cells, which is not so important in the sort of gene therapy context, but in terms of stem cell context, it may be more important. And this is an experiment with conditional mice in which we deleted the B7A gene, and the important point is that over time, one sees really a loss of chimerism from the mutated cells, indicating that B7A is required in some manner, as yet unknown, for really the maintenance of hematopoietic stem cells. So I think removing the protein itself may not be the best approach. So we think there's a therapeutic opportunity then for coming in with editing tools to disrupt the enhancer. This would obviously be done ex vivo, but this strategy would provide the sort of stable and maximal reduction of B7A only in the erythroid lineage, and we think the best reactivation of hemoglobin F that we can attain at this time. Now, how would one actually do this? One way, you could remove the whole enhancer, but is that really necessary? And we've been looking at this carefully, and this is the work of collaboration with Dan Bauer, in this case in the mouse, but similar work is going on in the human, where we basically saturated this region using, in this case, a library of guide RNAs for CRISPR-Cas9, done in a pooled manner so that one can essentially do a comprehensive saturation mutagenesis and enhancer with a reporter assay that we've worked out. But the bottom line is that in the mouse, the situation of the mouse, there's actually an Achilles heel within the plus 62 hypersensitive site with roughly this sequence, but we think it's a little broader than this, where essentially interruption of this region inactivates the entire enhancer function. It's equivalent essentially to knocking the gene out. So it's not necessary to remove the whole enhancer. We think a discrete cut at the Achilles heel will result in a similar, in a favorable outcome. And the same is true in the human enhancer as well, although the Achilles heel is located in a different position. So really the question then is, how can we leverage all this work for some sort of therapeutic benefit? We think the genetics now has come to the point where we believe it's reasonable to consider editing, particularly of the enhancer, to provide really a gene therapy strategy for increasing fetal hemoglobin in a permanent manner within individual. And I think most of the science has been done, save actually pinpointing precisely the optimal site at which one wants to make the editing. I think the challenge that lies before us, and the one that we're beginning to face now, is that if one is really going to impact these disorders, one needs a pill. In this case, removing the effective function of BCL11A in some manner. This is obviously challenging because it's a transcription factor, but there are a number of strategies one could think of using, attacking either proteins associated with BCL11A or protein interactions of BCL11A. But I think the development of small molecules would certainly revolutionize the management of all the hemoglobin disorders. And I think that if we want to set out one goal, that would be the goal for the future. So one of the questions that comes up is what level of BCL11A is critical? How much do you have to inhibit it in order to have enough fetal hemoglobin to actually make a difference? And genetics here has told us something very interesting and perhaps a little surprising. And so where do you want to be on this curve? There's actually a new syndrome that's recently come to light. Very rare patients have been described with deletions that encompass the BCL11A gene and sometimes additional genes, but sometimes only the BCL11A gene, on one chromosome. These individuals are unfortunate. They're ascertained because they have a severe autism-like or mental retardation syndrome variously described in the families thus far. And in recent genetic studies, actually, BCL11A has been associated in sort of large GWAS studies with autism and schizophrenia as well, indicating an important role in nervous system development. So this is a unique opportunity then to see what is the effect of having haploinsufficiency or 50% of BCL11A. What does it do in the context of hemoglobin expression? It's really quite remarkable. These are three patients that we've had an opportunity to look at. Two of them, Vijay Sankaran, and one I've obtained from collaborators in Canada. In one case, they involve only the BCL11A gene and a little microRNA here. These two involve another gene that's probably irrelevant there as well. But what's interesting about these patients is they have normal hematology in terms of red cells, normal lymphoid development, normal B cells, but their hemoglobin F levels are really quite remarkable, measured at 23%, 16%, almost 30% in a standard clinical lab. These individuals have 50% the BCL11A. In fact, this individual is known to have a high F SNP at the remaining BCL11A as compared to this individual, which I think is in accord with all the genetics. So we can imagine that really reducing expression, even on the order of half, 50%, would get the hemoglobin F concentration in roughly a range in which it would be therapeutic. So I think this is good news in terms of thinking about how to actually target the gene or the protein for benefit. So what are we trying to do now to get at this? Well, the BCL11A protein, as one might imagine, is actually part of many protein complexes. And within erythroid cells, it's found in association with transcription factors like GATA1 and FOG1. It's also found with the NERD complex, with HDACs, which bind at the N-terminus of BCL11A, and it's found in association with a number of other co-repressor complexes. In principle, any of these might be targets for therapy. We can go into detail later if one wants about specific candidates. One of the things we've been doing is trying to determine which parts of the BCL11A protein are really essential for its function, because we think that'll be important in thinking about small molecule development. And one of the curious features of this protein is in a rescue assay we can actually discard much of the protein, and it's still capable of actually regulating the globins appropriately. And the only critical regions thus far are the extreme N-terminus, which binds the NERD complex, this region here of two zinc fingers that actually also interact with GATA1 and FOG and SOX6 transcription factors, and the terminal zinc finger complex, which at least binds LSD1 and CoREST. maybe other things as well. We don't know if these are actually critical to function. But there are really only 3 parts of this large protein which we can ascribe to any essential function, which is curious if one thinks that one needs to form very specific protein complexes in order for the protein to function. And we're trying to understand the meaning of this at this point. We're using a number of approaches to try to develop small molecules that actually interact with B7A, at least as a starting point for further development. We're using a number of physical methods, which include small molecule microarrays, where one puts small molecules on a slide and uses recombinant protein with a tag to basically identify which small molecules bind the protein. One can do a similar kind of assay in mass spec. There are also larger versions of this with DNA-encoded libraries, or also other methods such as thermal denaturation. And we're using a combination of these methods, various libraries, with different collaborators to try to identify small molecules that at least interact with the protein as at least first hooks to really get at how it could then target the protein for therapy. And this is obviously going to be a long road. It's an uncertain road, but I think one that needs to be undertaken. We've run some pilots already with the small molecule microarray approach, and at least in the pilot screens, we can actually identify some small molecules, such as 2 here, that do actually physically bind B7A with a measurable affinity. And obviously, this is just a starting point, but at least it gives us some hope that this kind of approach with chemists and physical chemists will get us to some molecules that at least be tool compounds for subsequent study. Lastly, would having small molecules to B7A have any other role in therapy? Well, just this past week or two, as a paper appeared suggesting that B7A is actually a critical gene in triple-negative breast cancer, where it's proposed that it's required in the breast cancer stem cells of that disease. So at least one would have another avenue to pursue, for example, with small molecules, if in fact they prove to be, let's say, toxic for long-term use as they might be for hemoglobin F induction. So a few take-home messages in closing. We think B7A is a validated target for hemoglobin F silencing and reactivation. We think it's validated by genetics, biochemistry, cell biology completely at this point. Natural genetic variation in this erythroid enhancer modestly controls output of the gene, and they emphasize modestly. That's what natural variation gives you. But the enhancer deletion has a profound effect on expression, and I think this is an important and relatively underappreciated aspect relevant to GWAS studies in general, that very often it's assumed that if the GWAS result has a small effect size, it means it's biologically not very important in that entity, which may not be the case. We think that B7A is an excellent test case for therapeutic genome editing and engineering to benefit patients with the hemoglobin disorders, and we hope this comes online the next couple of years. And we're currently trying to identify selective small molecule inhibitors of function that might be used in the future for therapeutic purposes. And in closing, I'd just like to thank those who have actually done the work. Vijay Sankaran was an MD PhD student who reactivated the reactivation of hemoglobin F in the laboratory. Been followed by Jan Zhu and Dan Bauer, who have carried the project. Dan Bauer particularly on the enhancer with Sophia Kamran. And the chemistry work is being done by Jiang Hong, collaboration with Angela Kohler at the Koch Institute, and we have some biotech collaborators as well with various aspects of this work. Thanks very much, and I'll be glad to take any questions.
So active enhancers are bound by bromodomain proteins such as BRD4. I wonder, have you looked at whether BRD4 also binds on the enhancer of this, protein, is this gene, and whether, have you tried the JQ1 to treat it?
Yeah, you're asking whether BRD4 binds a lot of enhancers, whether the drug JQ1, for example, would be useful in this context. We've actually put JQ1 on, and it doesn't have much of an effect. So we don't think this is a BRD4 susceptible enhancer, at least in that context. And we've tried a number of other epigenetic inhibitors for various pathways that are known, and for the most part, those are relatively inactive.
Stuart, what are the prospects for reactivating delta? And the other question is, what is the role the methylation plays in control of gamma?
What about reactivating delta, which is sort of a minor delta gene? The delta gene is actually it's trying to become a pseudogene. It's kind of on its way out. So we don't actually think that's going to be a good choice. And I don't even know, some people may know in the audience, whether if the delta chain, if one made delta protein and put it in a hemoglobin tetramer, does it actually function well? I'm not even certain whether that's known. And the second question is methylation. Our view, and based on not a tremendous amount of direct data, is that methylation is really kind of a late event. It's after probably silencing has occurred, and particularly that if one uses demethylating agents in normal cells, one does get a little boost of F, but if you get rid of BCL11A, you get a lot more boost. So we think that actually BCL11A is the barrier to that as well.
Could there be some kind of a synergistic effect between use of hydroxyurea and knockdown of BCL11A?
Possibly, but we don't understand how hydroxyurea works, and at least in our work, they don't seem to be operating in the same ways. So
that might be good.
Yeah.
Can I ask you a question?
Yeah.
I'd like to ask a question. I believe that BCL11A occupies the LCR during both phases, since when gamma is active and when beta is active, and yet the interactions change between the LCR and each of those genes. So do you have any idea about what BCL11A is doing there?
Yeah, the question is between when gamma is expressed and beta is expressed, where BCL11A sits. We don't really know that. All we know is what happens in an adult beta-producing cell, that it's present there. We don't actually know if we knock down BCL11A, obviously the locus changes conformation, but we don't know where the protein is in the cells will express gamma. We think that there isn't much protein, so we don't think that it's doing much, but we don't know.
Okay. Oh.
Stu, in your mouse experiment where you had the Achilles heel,
Yeah.
that site looked a lot like some of the sites that EKLF binds to.
Exactly.
There are EKLF mutations associated with hemoglobin or HPFH.
Yeah, well, I think
how does that all fit together? That wasn't
the
part of
answer
your
to the
model.
last one is there are a few EKLF mutations which are associated with HPFH. We actually don't think it's an EKLF site, though. We've played around to try to see if EKLF is actually the offender there. We don't think so, and we think it's slightly bigger than just that site alone.
Thanks.
Well, if there are no more questions, join me in thanking Dr. Orkin for his lecture, and I would like to invite everyone in the audience to a reception and more conversation in the library, which is sponsored by the FAES, directly after the seminar.